So today I presented my data on NK cell phenotypes and MRD negativity in newly diagnosed multiple myeloma patients treated with Dara-VTD or VTD. And right now Dara-VTD is standard of care for newly diagnosed transplant-eligible multiple myeloma patients. And the primary mechanism of action of Daratumumab is through is ADCC via the Fc gamma receptor CD16 on NK cells. Therefore, it's crucial to know the role of NK cell subsets in Daratumumab-based therapies or combinations. So we included 152 patients treated in the Cassiopeia trial or the HOVON 131 trial, treated in the Netherlands and Belgium. And these patients were randomized to receive Dara-VTD or VTD, induction, autologous stem cell transplantations, and then 2 consolidation cycles of Dara-VTD or VTD alone. And if patients reached PR or better, they were randomized another time to receive Daratumumab maintenance treatment or to be observed in the follow-up period. We collected bone marrow samples at baseline, but also peripheral blood samples at baseline and during treatment. And we assessed NK cell subsets, activating receptors, inhibitory receptors by flow cytometry. And after manual pre-gating of the NK cells, we performed computational analysis to analyze our data. And what we found is that 64% of patients treated with Dara-VTD reached MRD negativity post-consolidation, compared to a third of the patients in the VTD arm alone. And in the Dara-VTD arm, the patients who had at baseline a higher percentage of the CD16, CD57 or DNAM-1 positive NK cells were more likely to reach MRD negativity post-consolidation. And we did not find this association in the VTD arm alone. And next we assessed the changes during treatment. And we found that for the Dara-VTD patients, the CD16 and CD57 decreased over time. And this was not as pronounced in the VTD arm. Besides, we found an increase of CD56 bright and NKG2A positive NK cells. Besides that, we also found that the absolute NK cell counts decreased over time. However, the patients that were treated with Dara-VTD, but after the second randomizations were in the observation period, in those patients we found that the phenotypes, so CD16 and CD57, the percentages restored, and this was also holds true for the NK cell, the absolute counts. So in conclusion, patients treated with Dara-VTD who had at baseline a higher percentage of the CD16, the CD57 and DNAM-1 positive NK cells were more likely to reach MRD negativity post-consolidation. We found that the phenotype changed over time, and this was more pronounced in the Dara-VTD group compared to the VTD group alone. And we saw that the phenotypic changes, but also the absolute counts restored after stopping Dara treatment. So this indicates that there is a potential for subsequent NK cell therapies after a CD38 targeting antibody-free interval.